This program is brought to you by Stanford University. Please visit us at stanford.edu.
Anyway, so what we did was we got some idea of how evolution worked and some idea of how we ended up with the biodiversity of life we have on the Earth. Now, we're going to take that and look specifically at what has evolved to be in the extremes. And the reason we're going to really focus on extremophiles for the whole rest of this lecture is because it gets back to these fundamental questions of astrobiology. Where do we come from? I've already suggested that thermophiles tend to tree out really early in the three domains, particularly in the archaea. And so there's a suggestion out there that life arose in a high temperature environment, although I said that it's more likely that they went through this high temperature bottleneck. So that's already an interest in where life came from, looking at the extremes, and are we actually all derived from thermophiles? And then the question of are we alone, looking out into our solar system and beyond into the universe. This is really important to understand extremophiles for because it gives us an idea of where life might arise and where it might be able to survive and evolve and so on. Now, I can't stress this enough. I'm going to give you lots of examples of cool little creatures that can do all sorts of weird and wonderful things. They can live in solid salt and they can live in freezing temperatures and all sorts of things like that. But I'm not once suggesting that any of these specific organisms will ever be found on Mars or Europa or Enceladus or anywhere outside our solar system unless we put them there. All we're doing is saying, okay, we have one data point for life, that's planet Earth. And if life can arise under those conditions in our first data point, we know there's nothing about these physical or chemical conditions that preclude something that we would call life in these same physical and chemical conditions around Alpha Centauri or Gliese 581c or d or whatever, because we know that it can occur on the Earth. So what we're doing is looking for an envelope for life by looking at the extremes on Earth. Now, when we go into the future of life, these extremophiles, the specific ones I'm talking about, might be very important. It might be that we can take some of these organisms like cyanidium, which is photosynthetic and can live at very low pH and high temperatures. Maybe that's one that we really literally could take to Mars, to terraform Mars, or something like that. So when we're looking life beyond the Earth, some of these specific examples may be extremely important. But in any case, the are we alone question is really where this extremophile business, is tremendously important. Okay, so that explains why we're doing all this. I think I probably showed this the first class, but we were going so fast that I didn't think it hurt to put it in one more time to remind you where we come from in astrobiology, that we're really looking at man's place in the universe. Copernicus started with looking at our place, the Earth's place in our solar system, and then Galileo, understanding the Copernican system, Darwin, our place as living organisms, particularly our place as Homo sapiens sapiens in the grand scheme of things. But now we're looking in astrobiology as our place, ours life on Earth in the universe. And so that's, again, where extremophiles come in. So what I'm going to do is mostly give you a lot of astrobiology about extremophiles, but I know that some of you might actually be interested in making money once you graduate from Stanford, so I will mention a little bit of some of the biotech potential at the end, for those of you who have not taken vows of extreme poverty yet. And we'll talk a little bit about space, which will be starting to get us psyched up for the space part of the course. So what is an extremophile? Someone hesitantly used the word extremophile the other day for their presentation. Extremophiles are in. It's considered the cool thing. So I've noticed an awful lot of people say, oh, I work on extremophiles. And what they mean is they went into a dark, damp cave, and it was very unpleasant for them, and so they said they worked on extremophiles. And I'm going to give you a much better definition, I think. Again, if I haven't mentioned this already, if you haven't looked in the syllabus, I would strongly suggest, and that's not a subtle hint, reading the article that I had in Nature a couple of years ago with Rocco Mancinelli, who'll be here next week, on life in extreme environments. It gets assigned to everyone else's classes all over the world, so why shouldn't it get assigned to you? A lot of what you see in here will be there, but not in as much detail. Some parts will be more detailed than what I'm giving you And some of this will be a little bit more current, because that was like only 2001. Okay, so what is an extremophile? Well, let's start with the Latin and Greek and be very, very scholarly here. Extremus is a Latin word. It means being on the outside, and you add to it philos. Who knows what philos means? If someone's calling in. If it's a Martian, we stop class right here. Philos is love. Bibliophile is someone who loves books. So therefore, an extremophile, you would think, would be something that loves to live on the edge. Now, this word was coined by Bob McElroy, who was at NASA Ames for many years, because even in the 60s, they were starting to do a lot of work on life from the hot springs of Yellowstone and other sorts of places like that, thinking that these might be good analogs for life if we found it elsewhere in our own solar system, like Mars and Venus at the time, and so on. It was, as you can see, a combination of Latin and Greek, and I was wondering if that was all right. Bob just passed away a couple of years ago. So I asked him, and he said, yeah, apparently the Romans did that all the time, too. So it's perfectly legal, and there you go. That's where the word extremophile comes from. So that all sounds very simple, but it is a little squishy in terms of definitions. So let's look a little bit more out of this. Do we mean physical or chemical or biological extremes? We always talk about physical and chemical. People don't tend to talk about biological. Maybe population density could be considered an extreme environment. I'm just throwing that out to start to broaden your mind's a little focus here. What do we mean by extreme? And these possible ideas that maybe it's just in the eye of the beholder, that a cold, dark cave really is extreme. Maybe there's some sort of evolutionary definition. Extreme is something that's farther away from the ancestral state, like we talked about horses' toes the other day. Or maybe there's some sort of objective definition. And I've sort of come full circle in my own thinking on this, and again, this is in that paper in Nature, that in fact there is an objective definition for an extremophile, and I will give that to you in a moment. We said extremophile means lover of extremes. Do they really have to love extremes? I mean, you don't go up to a bacteria and say, do you love living in boiling battery acid? No, it's not. In some cases, these organisms are exquisitely adapted to a particular environment, and they actually can't live outside that environment. So in a sense, you know, in terms of talking about love in a microbe, if you can do such a thing. I don't know if we have any philosophy majors in the room who'd like to discuss love amongst the microbes, but they love it in that they can't go anywhere else. But in some cases, we're really talking about organisms that can tolerate an extreme environment. All things being equal, Deinococcus radiodurans does not wake up in the morning and say, go ahead, whack me with 100 million gigarays of ionizing radiation. But if you do that, they'll make it through. Obviously, not gigarays, but I'm just making up a number. We'll see more sort of numbers later. But they can tolerate these extremes. So we do sort of have a whole range when we talk about extremophiles between ones that tolerate it and ones that really have to be in those conditions. And then the final thing, and this is a dirty little secret people don't like to admit, it's not like you get this little golden label that says, I am an extremophile, or I am not an extremophile. There are many organisms that in some points in their life cycle, they could be considered an extremophile, or under some nutritional conditions. And again, Deinococcus radiodurans is such a beautiful little bacterium to beat on because it's considered the poster boy or poster girl for extremophiles because it is so highly radiation resistant. But in fact, under some culture conditions, it's highly radiation resistant. And under others, it's not any better than anyone else. So it's a little bit of the condition it's under. I think an even easier example to get, particularly for those of us who live in temperate climates, are frogs. Frogs, some frogs at least, can freeze solid in the winter. Now, if you take a frog in July out of a pond, stick it in your freezer, slam the door shut, and come back a month later and put him on the kitchen counter, he's not going to go, sort of dust himself off and go croak, 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 jump, 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 and off he goes. He's going to be a dead, squishy, frog with all his membranes broken and so on. He wasn't ready to be shoved in the freezer. But by the late fall, when they've started to produce these antifreeze molecules and so on, you can take a frog of his sort, his cousins, or him later in the season, and put him in the freezer, and he will come out and go, you know, jumpity, jumpity, jumpity in the spring. So it very much depends on the physiology at the time in those conditions. Before I give you a real good definition of an extremophile, I put in this slide, why should anyone bother to study extremophiles? It turns out there are an awful lot of people who do. And one of the interesting things that I've noticed over the years is that really the study of the limits of living organisms can be tied very much in with food preservation. I'll show you what I mean in a moment. It's sort of a fun thing. Basic research, there are many extremophiles that it's, for various reasons, it's easier to do research on them than another organism. For example, you might have some compound that only dissolves at very low pH, so you use it on an organism that lives at low pH. Biological warfare, I don't need to say any more about that, but there's certainly potential there. There's certainly a lot of biotechnology potential, and I'll mention that at the end. There are billions, literally billions of dollars being earned today off the backs of extremophiles and extremophile enzymes. Certainly potential for future use in space. As I mentioned, we're going into harsh environments as we leave planet Earth, and so organisms that can deal with these extremes, these physical and chemical extremes, may be incredibly useful to us. But it also helps us to understand the biodiversity on Earth, origin of life. The archaea were first noticed not just because of the DNA, but because they were living in extreme environments. Mechanisms of survival, won't go into that too much, limits of life in the universe, I've already mentioned that. And the idea is everything there in that green box is a good reason to study extremophiles if you're studying astrobiology. Now, I would love to say this is all brand new and we were very clever and thought about this at NASA a couple of years ago and started on it. But it was pointed out to me by a wonderful historian of science in Canada that, in fact, Richard Proctor, who is a popularizer of science in the 19th century, sort of a Carl Sagan figure in the 19th century, wrote lots of books and so on and so forth, actually came up with this idea. In his book in 1870, Other Worlds Than Ours, he certainly connected the idea that we could take organisms that live in the deserts and so on the Earth and use these as examples for organisms that would live potentially on Mercury and so on. At that time, the solar system seemed much more habitable, partly because we just didn't know. And unfortunately, in some ways, it's become less and less habitable from what we know, and then it's starting to broaden out again. So at the time, he went through Mercury, Venus, and so on, his chapter on each planet. And it really is very much of an analogous approach that to what we use today. Where do we find these extremophiles? I just show you the same sort of three-domain graph that I showed the other day and circled a few other organisms that I would consider extremophiles. So the point of this is that they're all over the place. We could circle many, many more things that are not on this particular three-domain tree. But the point is that you see eukaryotic extremophiles, you see prokaryotic, you see bacteria, you see archaea. And the reason I stress this is in case any of you are under the mistaken impression that extremophiles are all high-temperature archaea. This is sort of a little bit of a rumor that's going around, and I'm going to completely debunk that myth and give you a much richer view of extremophiles. So the main take-home message here is that extremophiles are found all over the place. Okay, so now I'm going to really give you what is my best take at a definition for an extremophile. And that's very much objective. It's based on the fact that life is based on organic carbon, and we've argued that life anywhere in the universe is likely to be based on organic carbon. Now, if you're an organic carbon-based life form, your number one priority is not to fall apart. If you fall apart into little diamond flakes and carbon and so on and so forth, CO2 and carbon monoxide, you're not much of an organism, are you? So you have to keep this organic carbon together. So anything that destroys organic carbon is going to be an extreme environment. For example, high levels of radiation that can break backbones of DNA and lipids and so on, oxidation and literally breakage. It's going to be an extreme environment. The second thing is, since we're organic carbon-based, we have to also have reactions going on. And when we start to go to the extremes, just because of what we know about organic chemistry, enzymes are going to slow way down as it gets colder and colder, and they're going to speed up as it gets warmer to some extent, and at some point their activity is going to start crashing, and at some point the actual molecules denature. And what this means, for those of you who haven't had a background in chemistry or in elementary biology, if you're a protein, you're folded up. You're a chain of amino acids, it's all folded. As you denature, for example, like cooking an egg, this holds what's called secondary, tertiary, quaternary structure, unfolds. That's denaturing for a protein. For DNA, you've got a double helix. If it starts to unzip, unwind, you've denatured it. Okay? So denaturing means that it's no longer intact, it's no longer functional. So denaturing biomolecules is not a great thing unless you're doing it in a very controlled way as an organism. So any of these environmental conditions that denature biomolecules are going to be extreme. So again, this is a much more sophisticated definition than saying, oh, I went into a dark, damp cave and therefore I found extremophiles. No. Did that dark, dank cave challenge these organisms as organic-based life forms? Now, the reason we've said that life is based on organic carbon is that carbon is the fourth most common element in the universe. Llewellyn Mandela showed us that. And it's capable of forming a vast array of compounds ranging from methane to DNA, tons of organic chemistry around. Llewellyn Mandela pointed this out to us and showed even in the interstellar medium, you find this sort of thing. Now, that to me is one of the real clinchers, is that we find organic chemistry all over. It's not just planet Earth. That seems to be the language of complex chemistry everywhere. We don't see this huge array of silicon-based chemicals that do all sorts of weird and wonderful things. We can make a few, but nothing compared to the organic carbon, and we see it out there. And furthermore, I think Lou mentioned we're sitting on a large silicate rock, and we're not based on silicon. You know, there are days that I feel my computer is my brain, but the fact is that's the second generation of life, as Seth is going to say in his last lecture. But this is our first generation, the squishy organic carbon-based life form. So if you follow all that, life is based on organic carbon, and again, the extremes are the things that challenge an organism because they're based on organic carbon. This figure is basically an updated version of the one in the 2001 paper, and it's in color, so that's kind of fun. What it just does is gives you a list of a couple of things that we would consider extreme environments, and we're going to go through some of these in a little more detail. For example, temperature. Organisms that live extremely high temperatures are called hyperthermophiles. Just warm temperatures are thermophiles, and when I say warm, sort of the consensus is to be a thermophile you have to live between 60 and 80 degrees centigrade. What temperature does water boil at sea level? 100. You're pretty close to boiling. And that's just a thermophile. A hyperthermophile goes way up, past boiling. I'll show you in a few moments. On the other side are the psychrophiles. This is not psychotic phials, psychrophiles, lovers of cold. Okay? And so there are ones there that go actually below the freezing temperature of water. We'll talk a little bit about radiation and why that's so nasty. High pressure organisms, and here's an example where sometimes we take organisms up from the depths of the ocean, extremely high pressures, and they burst. They cannot deal with the normal atmospheric pressure. Others, if you take them up slowly enough, it's like, oh, okay, this is what it's like to live on the surface? Okay with me. So it depends on the organism. You cannot assume because an organism is from deep down in the ocean, from high pressure, that it cannot deal with the lower pressure. It may be barotolerant rather than a barophile. It's difficult to understand what the difference is between piezophile, pressure-loving, and barophile, which is really weight-loving, but those two words are kicking around out there. If any of you, by the way, are interested in more of these mechanisms and sort of more the technical details, I put it all into a paper that actually also is now in press. It was totally inappropriate for that paper, so I've had to take a lot of it out, but I now have all these thoughts and references together. So if anyone's interested, I'd be more than happy to email you that stuff so you can go into it in more detail. Desiccation, which is often tied in with salinity. We'll talk about, again, this lecture, pH, oxygen temperature, oxygen tension, chemical extremes. If you live in very high levels of mercury, you might well be find it's going to be a challenge. Let's be honest here. Organic solvents. We don't know organisms that can live in benzene because we're worried about that, or some detergent that would dissolve a cell membrane. I've reviewed papers of people who've made those claims, but they've yet to be convincing. That would definitely be an extreme environment because it would be nearly impossible to keep your membrane intact. You would have to figure out some way to get around this. Vacuum. I realize people don't tend to talk about it, but most organisms cannot walk into a vacuum and walk back out. Probably cockroaches can, and some beetles. They can apparently go into electron microscopes, have the vacuum pulled down, and walk back out. But most organisms cannot do that. So that I would consider that an extremophile. And then I started thinking, well, what about electricity, like electric eel? But they're actually protecting themselves from their own electricity. And then I really got carried away. I thought, well, what about organisms that germinate after a fire? And this is really cool. There must be seeds that are fire tolerant, and I'll figure out what the Latin word for that is and so on. Well, it turns out, for those of you who thought, oh, that's a cool idea, what happens is that the seeds germinate not because of the fire itself, but because of the smoke that's produced. Darn. So that one didn't make it onto the list. But keep thinking, because maybe you can think of some other sorts of extreme environments that are legitimately difficult or extremely impossible for a carbon-based life form to live under. Now, I get asked all the time to talk about life in extreme environments. A couple of years ago, I was asked to talk on the history of life in extreme environment research, which was fun because instead of trying to find the most recent papers, I was supposed to try to find the earliest papers. And that's when I started realizing that, in a sense, we've been working on extremophiles for thousands of years because of food preservation. And here's that same sort of figure, but now we're talking about food preservation. What do we do with temperature? Well, think about it. We boil things, we pasteurize them, we cook them, we refrigerate them, we refreeze them. What are we doing? We're trying to beat the odds with the microbes. We're trying to get rid of the vast majority, if not all of them, so the food does not decay. Same thing, irradiating food, putting food under pressure, desiccating it, drying it, or curing it. That's a very, very old method. Apparently the Romans just were wild about dried fruit, I understand. Salting, that's again a very old, very common method, salting fish, salting meat. High osmotic pressure, think about sugar preservation. The obvious example is jams and jellies. pH, very low pH, you could end up with sauerkraut. You can have some high pH, but I think it's more low pH that we use in terms of food preservation. Having oxygen-free atmosphere to retard food growth, food spoilage, and then chemical extremes. Ethanol, well, you wouldn't actually want to stick stuff in ethanol, but you can certainly preserve fruits in alcohol. There's no reason not to put something in brandy, for example. I believe, was it Napoleon's body who was sent home in a cask of brandy? I think it was, or Nelson. Anyway, one of them was sent home to England in a cask of brandy. So it's clear that we are well aware that alcohol is a good preservative. Smoking things works very nicely as well. So I just think that's kind of a fun parallel there. And we often have a lot to learn from other fields. So now let's go through some of these extreme environments. Are there any questions so far? Okay, so what I'll do is spend a few minutes, well, probably 20 minutes or so, giving you a little bit more of a flavor for some of these extreme environments, and give you some examples and some gee whiz, and feel free to say, wow, if the mood strikes you, it's a little like a revival meeting. Then we'll take a little break and then try to figure out what all this means. Okay, so let's start with temperature. Fun, don't you love keynote? Okay. Temperature, I mean, you can think, of course, I don't want to stick my hand in the fire, ouch, hot, it's going to burn me, and so on. But I thought instead we'll start with things that are a little bit more subtle. What does it mean to have temperature change? We feel chilly going outside, but there's a lot more to it than that. For example, the solubility of gases is very much tied to temperature. Now, you don't need to believe me. What you can do is go home and take a can of Coke and put one in the refrigerator or the freezer and one in a warm water bath in your bathtub, or boil it on the stove, put warm water around it, and then take them into the bathroom or outside and open them. And which one is going to go pow and which one is not? Any idea? Wow. Since we're sort of at that level today that either people are too embarrassed to say the answer, since I'm sure most of you know, go home and try it. Let me know how it works. And you will very quickly learn about solubility of gases and how it's related to temperature. Organisms have upper temperature limits because of denaturation. Not only do nucleic acids and proteins denature, but so do other things like chlorophyll, which denatures at about 65 degrees centigrade. So you don't find photosynthetic organisms above 65, at least not if they're going to use chlorophyll, because it simply is broken down. And again, to me, the easiest way to think about denaturation is thinking about frying an egg. You get this egg that has a white that's in essence clear, it's translucent, and as you cook it, it turns opaque and white. And that's because these proteins are denaturing. Okay? Same thing if you make ceviche, for those of you who like Latin American food, which I love. The way you're preserving that food, you're cooking it, denaturing the proteins, using acid. You're using lime juice or lemon juice or some vinegar. The same thing, those will also denature the proteins. So you've got these upper temperature limits because of denaturation. You have optimal temperatures for enzymes. And as the temperature gets lower, as I mentioned, things slow down, and at some point you actually get freezing. So the reaction rates are going to go down, 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 and therefore it's going to be harder to keep metabolizing and harder to stay as a living organism. Now, if you want to stay in a sort of suspended state, like an embryo in liquid nitrogen, that's fine. But ultimately, if you're going to be a real organism, you're going to have to reproduce and so on. So you really do need to maintain certain temperatures, at least periodically. And then let me give you another really nasty thing about going to low temperature, and that has to do with ice. Now, think about ice crystals. Think about what happens. Oh, here's a really unpleasant example. If you have ice cream that melts in the freezer, and then you refreeze it, what happens? You no longer have this beautiful, smooth emulsion that has gas and liquid and solid in every bite on your tongue, which is what makes ice cream so marvelous. What you've ended up with is ice crystals. And they're sharp little buggers, and you have no, as former advisor said, you have no mouthfeel left. It's not a pleasant experience. You throw it out. Well, imagine these ice crystals forming not in your ice cream, but within a cell. Shrink yourself down to the size of, say, the nucleus of a cell, and imagine little ice crystals forming. These are like little swords going right through the membranes. And so as the temperature goes back up, as you defrost, you've got a whole lot of leaky bags, leaky water-filled balloons, and so therefore you're going to die. So ice crystals are incredibly hazardous to life. So if you're going to freeze, you're going to go into liquid nitrogen or whatever, you have to start playing tricks so that you're not forming ice crystals all over the place.
That's exactly what I was going to ask. How do you do that?
Antifreeze. And I'll show you also another way insects do it. They can use ice nucleation sites. But I promise. Okay, so temperature, there are good reasons that temperature is like ground zero for extremophiles. And this is just, again, to remind you that temperature does matter. I'm not going to spend any time really on this slide and the next slide, but again, this information is nowhere else without you having to do a lot of searching. So I just put it there if you want to go read it later. A lot of this real interest in high temperature research started with Tom Brock, who was a young professor at Indiana at the time. He's mostly retired now from Wisconsin. But he was actually doing other sorts of work, and someone had suggested he go to Yellowstone. And he was one of those who didn't say, well, everyone says it's not, organisms won't live at these higher temperatures, so I won't bother to look. He started to look, and lo and behold, discovered that there were living organisms in some of these extremely hot places. Now, at that time, 60 degrees centigrade was considered hot, and it is hot. I mean, that you don't want to take a bath at 60 degrees centigrade. but compared to what we know today, that's nothing. So he started finding these living organisms and published a big paper in Science in the late 60s. I think it was 67. Anyway, I've got the reference for you if you want it. And that really got a lot of people excited. I've had older people in other fields of extremophile research tell me that's how they really got excited about working on extremophiles, is Tom Brock's paper. So it really was a seminal paper in that regard, and being in Science, it was a very short paper. But it's really quite interesting. He then spent many years with an army of graduate students and undergraduates at Yellowstone, setting up trailers and so on, and doing incredible research in many of the hot springs there. In the meantime, there's Alvin and other sort of deep sea vessels that NSF has sent down, the National Science Foundation, and they started in the late 70s discovering whole ecosystems in these hydrothermal vents. So these hot smokers, think of them as geysers that are underwater. And so again, here were places that no one imagined life could live. And yet, here were these incredible communities that weren't just microbial, worms and other sorts of invertebrates there as well. Okay, so what do we know about the temperature limits for life? Well, we're right there sort of in the middle. We're sort of in that boring mesophile area, sort of like with mosses and vascular plants and other, on the more boring side. Now, the upper temperature limit for a eukaryote is about 60, 65 degrees. So, you can push it a little bit. And you can certainly push it for very, very brief periods of time, but very brief. So you've got fungi, algae, protozoa that start maxing out there. I think I have a picture later of a fish that I found at 65 degrees. But you're really sort of pushing it there. Now there's some cyanobacteria that can go a bit higher. And then those tend to drop out, and you've got the heterotrophic bacteria. And then at the very highest temperatures, and now we're blasting through above the boiling temperature of water, there are some archaea. And this is the one example where I will freely admit the archaea are the champs. There are reports even higher than 121 degrees centigrade, and again, there's no other word for it except this is damn hot. This is way over the boiling temperature of water. This is not where you want to spend your life, and yet there are organisms that do that. And they've really had to do a lot of engineering to keep themselves humming along at these temperatures. I say there have been unconfirmed reports, even higher temperatures. You will no doubt periodically see these claims in the paper. Some of them will turn out to be true, I'm sure. And in the meantime, most of the other ones are unconfirmed. And so the firm number we'll leave you with right now is 121 degrees. Now, notice I have the Pompeii worm there at 70 degrees. This is something that was discovered in one of these around the top smokers that the submersibles went to. Well, this is kind of interesting. Who here has seen any, and I'll give you credit for any, of the Harry Potter movies? Any? Is there anyone here who has not seen a single Harry Potter movie? Okay, you got an assignment this weekend. All right, you are aware that Harry Potter exists. You're aware that there's a giant named Hagrid in these. We're together so far. Okay. Big, heavy costume. Now, I understand that the way this actor is able to deal with this is that they've actually run tubing of cold water under his costume to keep his body cool during the filming. That is exactly what the Pompeii worm does. They're right there on the edge of these smokers, and so they're sucking in colder water away from the smokers and running it by their bodies inside these tubes. And so, in fact, their bodies are not feeling these very high temperatures, but they are able to live in these high temperatures because they're running a cooling system through. Call that cheating, if you wish. I personally don't. I have had many scientists tell me, though, that's cheating, they cheat. Well, there's no book of cheating for an extremophile. This counts, guys. This counts as far as I'm concerned. That was very clever. They should get a little gold star and not be called a cheater and sent to the back of the room. And on the other extreme, I can talk about the Himalayan midge for about 30 seconds, probably not even that long, but I understand that it's active at minus 17 degrees. There's certainly plenty of fish in the Antarctic that are good down at about minus 5, and they are ones that do actually synthesize antifreeze molecules for their blood and their cells and so on. That's fine, but there are ones that blast even further. And then, wait, since we're on movie day, how many of you have seen the March of the Penguins? And these poor little creatures, they're clearly living well below zero. And again, I've had people tell me, oh, but they don't count because they've got feathers, you know, and they've got fat. But, you know, polar bears have fur. Well, again, there's no chain, there's no guidebook, excuse me. It says here in, chapter 3, paragraph 4, that if you have feathers, you don't count as an extremophile. I think they do. Why should a feather not be considered a perfectly good adaptation, but a molecular one, counts, or antifreeze counts or something? So if you look at it that way, the range is actually phenomenal. And as I mentioned before, we certainly have seeds and spores and even human embryos that can live in very, very cold conditions, liquid nitrogen. But they don't spend their whole lives there. This is a dormant stage that you then have to be pulled out of. So let's look at one of these environments with very hot temperatures. This is Octopus Springs in Yellowstone. How many people here have been to Yellowstone? Okay. Yeah, Octopus Springs is a little bit north of Old Faithful. So it's in a part of the park that tends to be about pH 7 to 8 in terms of the water chemistry. This is considered Mecca for microbiologists. The source here is over 95 degrees centigrade, so it's over the boiling temperature of water for Yellowstone. So you can sort of think of that as the body of the octopus, and then the tentacles that are coming out here to the right are the outflow streams that get cooler and cooler. Now, cooler, once again, is a relative word. Here's this little outflow here that's up in the 80s. Again, this is extremely hot. I accidentally stepped in it once when I was really tired having worked there all day, and you have never seen anyone take a boot off so fast in your life. 80 is not cold. It is hot, 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 hot. Okay? And yet there are organisms living in there, these pink, mushy things, Thermocrinus ruber. There are even organisms in this source. Now, when you start to go into some of these other outflow streams that are down at nice 65 degrees C, which is still hot, 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 you end up starting to get photosynthetic organisms. So you see that beautiful mat up in the corner. It looks like sort of maybe a layered jello from hell or something. On top, you see the green cyanobacterial Synechococcus. Okay, so it's just being able to make it. The chlorophyll's just on that borderline temperature. In that middle picture, you see that one of the outflow streams, and you can see without even a thermometer where the temperature gradient is. As soon as that temperature is below about 65, you see green. Okay? So the organisms that are above 65 can't be photosynthetic because of the problems with chlorophyll. So there's a little tour through Octopus Springs. And let's move on for a moment to pH. Who remembers what pH is? Ah, we've got a brave volunteer in the back. Or you just wanted it noted that you remembered what pH was?
We indicated that.
We indicated that we remembered having learned it. It's stuff about log negative, log the concentration. The best thing is just think of it as a concentration of hydrogen ions. Okay, so low pH, you got a lot. High pH, you don't have very many. That's sort of the simplest way to think about it. Now, we all learned in high school or even younger that the pH scale goes from 0 to 14, and 7 is neutral in the middle. Actually, rainwater is a little more acidic than that. But you know what? It ain't necessarily so. If you take that scale and you then look at the equations, negative log of the concentration of hydrogen ions, you'll realize that you actually can go below pH 0 and you can go above 14. And they're now starting to sell more and more pH meters that will go from, say, negative 5 or 6 or lower up to 16. So pH scale is actually even broader than 0 to 14. That being said, most of us live in a pH 7-ish world. Who has an idea what the pH of the ocean is at the moment? The average pH of the ocean. Oh, this is one of those classes that's great for cocktail parties. 8.2. Okay, with CO2 going up, there have been predictions that the pH will go down as much as a half to a pH unit in the next 100 or so years, which is going to be nasty for organisms out there. Okay, so the ocean is about pH 8.2, so that gives you some perspective. Now, there are fish and so on down at pH 4, and flies and so on down around 2. You're now starting to talk about things like vinegar and lemon juice. When you're starting to get to pH 0 to 1, where you've got things like sulfolobus, you're basically talking about battery acid. Sulfolobus, though, does a double whammy. They were originally isolated from Congress Springs. This is in the Norris Geyser Basin area of Yellowstone. So it's quite a bit further north than those other pictures I showed you. So it's extremely low pH in that area. And this particular spring is boiling temperature. And yet, that's where sulfolobus lives. So sulfolobus, the bacterium, lives in boiling battery acid. And that's where it actually likes to live. It takes all sorts to make a village. There are other organisms that we found down to pH 0. And as I said, and including an alga, there's Cyanidium caldarium. You see those little round things. You'll see those again because they're one of my favorite little creatures. We're going to start to find things below pH 0. Jill Banfield at Berkeley has been one of the leaders in this field. She's been doing a lot of work in acid mine tailings and has seen these organisms down at 0 and say, I think she's starting to push down to the negative numbers. Very cool. If you look at the opposite extreme, at the very high pH, where there aren't many hydrogen ions, you really want to go to the Rift Valleys in Africa, and that's where I took the picture down below of the flamingos. That's a lake that's up at about pH 11.5, and there are plenty of things there. As you can see, there are humans in the background, there are flamingos, they're chowing down on cyanobacteria, there are some crustaceans in there. There's not a tremendously rich community, but it's certainly got a broad taxonomic distribution. And so you've got all these organisms that can live at pH 11, 11.5. There are lakes out there, and we're talking now in the Rift Valley of Kenya and Tanzania, or as they say in Kenya, Tanzania. Some of these are up around 12.5. It is, I've been told impossible, but I try never to say impossible, nearly impossible, to keep a culture medium in a lab bench at pH 12.5. But yet we know that there are living organisms in these lakes at 12.5, so we know there's life that can live at least 12.5. And we may well find things even higher, but that seems to be what we've found so far on planet Earth are the upper limits. So think about that. Life from 0 to 12.5, and maybe even below 0, maybe even above 12.5. I mean, that just blasts apart the sort of world that we're living in. Salinity, very high salt, okay? French fries and all that sort of stuff. Again, it's difficult because if you think about it, proteins have to worry a lot about electrostatic interactions. So do nucleic acids, and you start messing around with salt concentrations, you got problems. You've got problems with osmotic pressure. A couple of years ago, I did a cute science fair project with my daughter, who actually hasn't been in yet. And what we did was we looked at what happens when you add high salt concentrations to something with a semi-permeable membrane, like your cheek cells. And it was very cool. You could actually take salt water and mix it with cheek cells and look under the microscope and watch them shrinking. And so, of course, being a kid at the time, she's still a kid, but say, 10-year-old, what better thing than to try a real-life simulation, chowing down on Fritos or, potato chips and checking again, and sure enough, when you do that, your cells shrink in your mouth. And vice versa, when we have a certain salinity in our saliva. So when she added water or drank water and washed out her mouth, some of the cheek cells lysed. They exploded because they took in so much water to try to keep the osmotic pressure even. So it does actually work in real life, too. Salinity matters. There's a lot of thought that we meaning life, arose in a saline environment similar to the ocean. I had a wonderful master's student from the geology department here last year. We were looking specifically at that. Can you take clues from the cytoplasm of cells to get an idea of what the primitive ocean might have been like? And so salinity, we are very much tuned to a low certain salinity. But that being said, there are halophiles, and these are primarily bacteria and archaea, although there are one or two eukaryotes, Denaliella salina is the one in particular, that can stand very high levels of salt. And when I say high levels, I mean they can live on solid salt. It's unbelievable. That top picture that's red is from a salt concentration pond at Exportadora de Sal, which is about halfway down the Baja Peninsula. It's the largest salt company in the world. As you can imagine, the Baja Peninsula, it's dry, it's hot. And so they take in the water from the ocean and it goes through a series of evaporation ponds. This is one near the end where the salt's actually going to crystallize, but not quite at the end. You can see on the edge there's solid salt, there's some foam from broken organisms. But the most striking thing about that picture is that it's red. Does anyone have any idea why that pond might be red? Okay. I will tell you, because this is another wonderful little piece of tidbits that you can pull out at a cocktail party sometime. It's red because halophiles, organisms that live in high salt, almost invariably have very high levels of carotenoids, which are red-orange pigments. Okay? So when you fly over the bay in San Francisco until Cargill completely disbands, you see these red salt ponds. And next time you see those, you fly over, you sort of calmly nudge the person next to you and say, oh, it's full of halophiles. Say, how do you know? Carotenoids, it's high salt, everyone knows there are halophiles there. And so, you can actually do microbiology from an airplane. Trust me, virtually any time you see something red like that, it's because it's high salt environment and it's because there are halophiles in there with carotenoids. Now in the bottom picture, what you see is a salt crust that's got some gypsum in it besides halite, besides sodium chloride. And you see a green band in there. Those are cyanobacteria. We've taken some of these organisms that actually are metabolizing in there. They're not, dancing around, but they are metabolizing. And we've flown them in space. And again, Rocco and Gerda will probably talk a little bit about that next week. And furthermore, they've survived. So there are a few organisms that can beat these odds. By the way, Denaliella salina, say, is the one that's eukaryote. It's actually green alga, but it has a lot of these carotenoids, so it looks red. It's a little more orangey than a lot of those others. But it's interesting, one of the ways that it deals with a high salt environment, besides making a lot of carotenoids, is also making a lot of glycerol. And so it can be a commercial source of both the beta-carotene and the glycerol. And there are some countries, particularly places like Australia and Israel and so on, that commercially grow Denaliella salina just for those reasons. Now, desiccation is very much tied in with salinity tolerance. Organisms that can deal with high salt can also deal with desiccation. Not every organism that can deal with desiccation can deal with high salt. It doesn't necessarily work both ways. But if you think about it, it's not great to be highly desiccated because you've now concentrated all those solutes in your cell. And so it's going to be much more difficult for enzymes to react. And if you're desiccated enough, if the water concentration is low enough, you're going to go into a state where you can't metabolize at all. You're just sort of hanging out, waiting for water. Now, imagine that you're doing this, and you're, say, this plant up down here at this Inca ruin, 15,000 feet. So you're up around 5,000 meters in altitude, where there is a ton of UV radiation. You're getting whacked, and you can't do a thing about it because you're desiccated. When you finally see those few drops of rain, which eventually will come, your number one priority is to go in and fix all that UV-created damage that has occurred while you were desiccated. Number two is to get, kick on the photosynthesis and reproduction, all those sorts of things. But these organisms tend to not only be desiccation resistant, but therefore also radiation resistant, because they have this problem of accumulation of damage while they've been desiccated. There may be a few organisms that can live on water vapor. There are organisms that can grow on, highly desiccated conditions. They're not growing, but they can at least hang out in those conditions for many years. And certainly there are many spores and seeds and so on that can do that. So I'm going to finish up this part with the radiation exposure and then let you stretch for a few minutes. I'm sorry to go over, but it makes it a more logical break. Radiation, we have different kinds of radiation, of course. Some, like ionizing radiation, creates ions out of molecules, and they've been around for many years. Other sorts, like uranium mines, of course, but other sorts vary seasonally, diurnally. I'm thinking of UVB in particular, UVA. Exposure is going to depend on the ecology where you live. If you're living near the surface, you're going to have to worry about solar radiation much more than if you live in a trench at the bottom of the ocean. We now have man-made radiation levels, so we've got new conditions, new environments in nuclear reactors and so on that organisms might like to give a try. And then we have radiation that we don't see on the surface of the Earth because we're protected by our magnetosphere or by our atmosphere that an organism beyond Earth would see, whether it's an Earth organism going beyond Earth or any other potential life form. So radiation should be incredibly important. But radiation is also important if we look at it from the evolutionary point of view, because natural selection, which you can see we're working our way up to Darwin's birthday here, has two basic components: generating variation that will be then passed on to the next generation, heritable variability, and then having selection among these variants. And radiation can do both of these things, so it ought to be right in there as an important extreme environment. As I mentioned before, radiation can also break or certainly modify proteins, lipids, nucleic acids. None of these things are great for you. I started to show you something like this the other day when we started to talk about radiation. Here's the solar spectrum down below. We live in a very tiny slice of that, right in there between the UV and the infrared. That's our visible world, between about 400 and 700 nanometers. That's the world we experience. That's also the world that plants operate in. The vast majority of plants are using light between about 400 and 700 nanometers. There are some bacteria that can use longer wavelengths, but what's going to be a problem with using a longer wavelength? Not that much energy. So you're going to have to really need to use that longer wavelength if you're going to go into the infrared. There are some snakes that can see in the infrared, and if you're a snake, like a python, that's a particularly great thing because you can pick up prey at night. You can sense their heat and use that as a visual clue. Okay? And then there's certainly lots going on in the UV, but as I mentioned before, and this, by the way, is just a spectrum I did outside my window at Ames last year, so it gives you an idea that we don't have a flat spectrum. We get more in different parts of the solar spectrum than others. As I mentioned before, proteins have a peak absorption of 280 nanometers, DNA and RNA at about 260, 259, and so any radiation that's anywhere near there becomes extremely dangerous if you're based on organic carbon, as we all are, and as we think all life in the universe would be. So this should certainly be an extreme environment, as with temperature, pH, and so on, anywhere you go. This is not an Earth-centric view. This is life. And to just give you, I'm going to show you one data slide because I think it's such a beautiful example of how important UV is, even on the modern Earth, even in March. This was an experiment that a wonderful master's student, Jackie Gargay, did with me a couple of years ago. She's now back in the UK at Cambridge University Press. What she did, she got up early in the morning. I can't take credit for it since we're being filmed. I would normally take credit, but I'm going to give her full credit. She got up early in the morning. She took some yeast cells and divided them into 3 different categories. Some of them, they all went out into the roof, so they were exposed to natural solar radiation in a water bath, so they stayed the same temperature. Some of them were put under UV transparent plexiglass, so it's just plastic that you could see through, but UV could also get through. So it just looked like a clear piece of plastic. That's what's marked UVT. Some under OP3, which is UV opaque plexiglass, so we could see through it, but the UV could not go through. And some under Mylar, which blocks the UVB, but not the visible or the UVA. And she just let them rip. And you can see that even by, even by noon, you could see a huge variation in the growth rates. This is March. This is not the middle of the summer. So you can just see from that what a huge influence UV has today, even in March in the Palo Alto area. Okay? I'm sure every one of you has experienced sunburns before. We know the UV's out there. We know that it's out there. But it gets worse than that. It turns out that UV can create reactive oxygen species. We actually create reactive oxygen species too because we're aerobic. What happens is when we metabolize, we take in oxygen, of course, and it's used as an electron receptor. So it ends up being reduced to water. Now there's nothing wrong with water. There's nothing wrong with oxygen. It's all these other radicals in the middle, particularly this hydroxyl radical, which I've got the pink star behind, that are nasty. And again, these do a job on your lipids, on your nucleic acids, on your proteins. Reactive oxygen species are nasty little creatures. And these are produced by not just UVB, but also UVA. So whereas UVA may not be so much of a problem for an organic carbon-based life form as a direct damage, indirectly through the production of reactive oxygen species is nasty. And so the conclusion that you have to draw is that we're extremophiles too, because we have to deal with this high oxygen environment. I just want to say, of course, I think that it's wonderful that we're aerobic, because if we weren't aerobic, we wouldn't be here. It's allowed the evolution of eukaryotes, as I mentioned on Tuesday. It's certainly allowed large organisms. We couldn't be anaerobic and be like we are, and we certainly couldn't have these incredibly high metabolic demand organs such as our brain if we weren't aerobic. So it's a wonderful thing being an aerobic organism. And oxygenic photosynthesis is wonderful. Yeah, life is really, it's all wonderful, but it's a dangerous game we're playing. We're literally playing with fire because we're burning things. And so I'm going to leave it like that for 3 or 4 minutes and let you stretch, and then I'll give you a few clues to how these organisms have figured out how to beat the odds and being extremophiles. If you're really interested, I went through each of these extreme environments and why it's so dangerous, and what we can do about it, or what these organisms do about it. And I'm only going to give you 2 examples, but if you're interested in going into more detail, just email me and I'll send it on to you. Okay, this is the first time you'll see this slide. I've heard it called Lynn's laziness principle before. This will probably be in my tombstone. To put it more technically, life is lazy, life is efficient. You don't do something if you don't have to do it. This is something that I'm sure resonates with all of us at a gut level. You don't do it. If you go to, you're at home and you go down to breakfast thinking, well, I'll, make myself a scrambled egg and some toast and a cup of tea or something, and you've discovered that your mother's already done that for you, you don't make another one just to prove a point. You take what's there, okay? So organisms do that too. If they're going to push the extremes, and remember we've seen this all over the taxonomic realm, and so therefore this is a convergent feature. We see organisms over and over trying to do battle with these extremes. They're going to do the easiest thing they possibly can do. And to me, the number one easiest thing out there is dealing with pH. Now, again, think about it. pH, low pH means there are too many hydrogen ions out there. And yet, if you let them all in, all these little ions, you've really screwed up the cytoplasm of your cell. You've now really messed up the electrostatic interactions with your nucleic acids, with your proteins, with your lipids, and so on. It's not a good scene. And as we mentioned before, those of you who are familiar with making ceviche, you can actually use the acid to denature proteins. That's what we do when we make sauerkraut. So you don't really want to let all these guys into your cell. So the organisms that live at very low pH all have converged on the very obvious solution, and that is you keep them out. You make a hydrogen ion pump. You just keep failing if you're in a leaky boat. You get those out. That means you can keep your cytoplasm at a nice comfortable pH 7-ish, and you don't have to do anything to the internal components of your cell. All you have to do is maybe fix a few proteins that are normally on the outside of your membrane. So that's very, very simple from a grand scheme, and that's why you see organisms in a couple of different taxonomic groups all figuring out how to make proton pumps. Well, there's actually a lot of precedence for proton pumps, but to use them to keep their cytoplasm at a relatively neutral pH. Okay, and the other way, if you're an alkylophile, if you live at very high pH, you need to, again, keep your cytoplasm about pH 7, so you concentrate those ions in there. You don't want to mess up, your cytoplasm if you don't have to, because then you're going to have to start going through and engineering all your nucleic acids and your proteins and so on. You don't do that if you don't have to. You keep your internal environment as close to what it was as possible. Now, obviously, this does not work if you're an Archean living at 121 degrees centigrade. You're going to have to do an awful lot of engineering to keep your proteins and your nucleic acids and so on from denaturing. And we know a little bit about it. There are a couple of tricks that are used, particularly with RNAs for stability. And again, it's in that paper that I'd be happy to send around to any of you that want to see it. Yeah?
Does this require a lot of extra energy?
Yes, but maybe the trade-off is worth it. You've got a whole niche to yourself. You've left the rat race. It's, I'm going to give a silly example. You know, maybe you don't like living in Palo Alto. Maybe there are too many people for you. Well, you know, there are a whole lot fewer people in the Antarctic. It's going to take a little more work to live there, but if you like open spaces, it's a great place. You don't have as much competition. You're more directly against the elements. This is the one other example I was going to give, and that's on the freezing. Again, there are a lot of fish that produce antifreeze, literally glycerols and so on, in their bloodstream, but what some insects do is a little different approach. What they do is create ice nucleation sites outside of their cell, so the ice crystals form outside of the cell and therefore don't rip through their cell membranes. And so here's that whole idea diagrammatically. Again, cute little tricks. It's as little as possible that you can get away with, because you really want to spend your energy on something else, and the something else is obviously eating and having babies. So that's, you know, pretty much life. That's the biologist's view of life. You eat, you have kids. You eat, you have kids. You don't want to spend a lot of time fixing your membranes if you can avoid it. Now, what about UV? I was talking during the break, someone up here. We have enormous numbers of ways to deal with UV and oxidative damage. I remember reading somewhere that the estimate is that every cell in our body gets 17 oxidative lesions, so damage 17 places in our DNA in the nucleus of every cell every day from oxidative damage, and that's not doing anything. This is for your average couch potato. If you want to, you know, like you're watching a Super Bowl, you actually get up and you want to go get something to drink, or, you know, more potato chips or something, your 17 number may be going up from there. So this is something that we are dealing with constantly. We don't even realize it, but we're aerobic organisms. We have to deal with oxidative damage. We have to deal with UV damage. And then I mentioned, for those of you who've never heard of it, there is a defect that's known among some people called xeroderma pigmentosa. And these people, who are unfortunate people, do not have the ability to do excision repair, which is one of the methods that we have to fix both oxidative and UV-created damage in our DNA. And they are exquisitely sensitive to sunlight. They basically cannot go outside during daylight hours. You imagine these kids, well, when they're kids, are going out and using playgrounds at midnight, that sort of thing. That's how sensitive we are if we don't have all these repair systems. So like I said, we're extremophiles. We just don't think about it because our bodies are doing this constantly. But we're having to deal with these two extremes constantly. But lots of other organisms have other tricks. Pigments are great tricks. You know, we use those in the, looks like most people in this room are adjusting their pigments in the summer in response to the UV. And there are certainly organisms like these algae down here do this. You can see these purple pigments. Those are the older Zygogonium who are making this UV blocking pigment. The juveniles, or the green ones there, they haven't figured out they got to start making this pigment if they want to live in the sun. We already talked about organisms with a lot of carotenoids, and you can tell in that picture in the upper right, it's sort of orangey red. You know right away it's a high salt environment, and the halophiles there, because you're all much more sophisticated than you were an hour ago. You know that you can take antioxidants. You know, you see these great vitamin bottles in health food stores and so on. And that will help scavenge some of these oxygen radicals. I am not in any way suggesting you run to a health food store. There is no evidence that it actually works beyond in theory in terms of human health. But that's what those do. They scavenge reactive oxygen species. We repair the damage. I mentioned xeroderma pigmentosa, what happens when you don't have some of these repair methods. Okay, so I'm going to avoid spending the whole rest of the time on DNA damage repair. And what I'd like to do is maybe someone you want to turn down the lights a little bit? See, I've proven that I've memorized one name so far. A little better than that, but not much. Thank you for sending stuff, everyone who has. Let's do a little, field trip in room 140 here. I'll give you some examples of some sites that are extreme environments: geysers, hydrothermal vents, ice, polar regions, subsurface, high altitude, high salt, and so on and so forth, mine drainage. This is a little bubbling hot spring by the road in the Norris region in Yellowstone. This is Iron Springs, bubbling, boiling hot, pH around 2, sounds cozy, and very high iron, and yet as soon as the temperature's right and so on, you see living organisms. And I just wanted to tell you that this actually fits in a lot with what I do in my day job, because of the whole idea of what the early Earth might have been like and what the limits are for life in the universe and going beyond the Earth. So because of that, the field trip I'm going to take you on, of course, is somewhat idiosyncratic. It's going to be my field trips. These are some of the places that I've been in the last 15 years or so. We've done certainly some work in Lassen and San Francisco Bay. I was very bad. I didn't put the state of Maine up there, or Rhode Island, where we've certainly done some work. We're doing more and more in the state of Maine on some of their leftover mine sites. Yellowstone, that got shifted way over. Before I post it, I'll move Yellowstone back where it belongs. It obviously is not in the Pacific Ocean. It is in Wyoming and Idaho and Montana. And that's wonderful, obviously, for these high temperature and low pH environments. Baja California, whoa, everything shifted a bit there, is also not out near Hawaii. That's that little thing sticking out down south of California, and that's where we see these very high salt environments, the salt companies. The Bolivian Altiplano, I'll show you very high elevations, very high levels of radiation. Kenya, it's got these very high pH environments. Hot springs in New Zealand. Again, New Zealand seems to have, everyone seems to have shifted today. Australia, fortunately, the name's big enough that Australia is on Australia. We'll show you a site there where we have some cool little ponds and Flinders Ranges. So let's just start close to home. Here's a nice satellite image, Landsat image of the San Francisco Bay region. And in the southern part of the bay, you see the Cargill Salt Company. It's been out there for over 100 years. As mentioned earlier, it's decommissioning. And so you won't see those red salt ponds forever. But it's been a really nice study site in the meantime. In fact, my wonderful research associate, Dana Rogoff, you see in the picture there, outsampling is going to be out there tomorrow collecting as well. So it's a great site in the meantime, but it won't be there forever. We can also close to home find psychrophiles. If you go up to Lassen, even if you go up in July, which is when these pictures were taken, you can see patches of snow some years. And you see that the snow looks a little discolored up there. You see it looks a little pinkish. And that's because of what's called watermelon snow, because of the color. And this is usually, at least in this area, Chlamydomonas nivalis. Again, it's a little green alga, lots of red pigment, lots of these carotenoids to protect it from the UV. It's not because it's high salt there, it's because of the high UV. And so you end up with pinkish snow. There are a couple of other algae that also live in snow and are also considered snow algae, but this is the common one around here. So and actually it's fairly common throughout the world. So those are psychrophiles close to home. If you haven't been to Lassen, it's only a few hours north from here, and it's a great little park. There's a picture of Chris McKay over on the left, getting ready to jump into a hole through one of these ice-covered lakes. I think someone talked about them the other day in the Antarctic. Chris has actually been swimming down under the ice, and he'll be in a few weeks to talk to our class. If you want to talk to him about that, you certainly can. What's marvelous is that these things are not frozen all the way down. On the bottom part, it is liquid. And you can see in the very bottoms these really beautiful microbial communities. So you immediately think, wow, that would be a great analog for life on Enceladus, on Europa, where you've got ice-covered oceans, we think. So we have a great set of extreme environments there that are also beautiful analogs. Let's go back to Yellowstone for a second. This is one of my field sites, Nymph Creek. And again, it's in this Norris Geyser region, so it's pH about 2 to 3. And here's this little stream. It's only a few centimeters in depth, nice and warm because it's fed by a lot of little tiny geysers. So the temperature's around 40 degrees centigrade, and that's pleasant. You know, 80 is not pleasant. 40 is very pleasant. And what you see is almost a pure culture of Cyanidium caldarium, which is a red alga. I'm not colorblind. It's a red alga that just doesn't happen to have a lot of the red pigment that it would other red algae have, phytoerythrin. So it looks green because you see the chlorophyll there. But it is a red alga. That picture up there on the upper right is a micrograph I took. That is not a culture. That's a field sample. So you can see what I mean by it's pretty homogeneous. There are a few green algae in there, some Chlamydomonas, there's some Dactylaria, it's a fungus, but it's pretty much uniform there. It's really an amazing place to work. But again, you're 40 degrees centigrade and pH 2 to 3 or so. We've been to places in New Zealand, same pH, same temperature, you end up seeing a lot of Cyanidium, or its close relatives, Galdiera and so on. And my feeling is Cyanidium can grow pH 7 fine. It can grow at room temperature fine in the lab. But if it's a mixed culture, it gets overgrown by other organisms. So what I think is happening is the reason we find it in these places that are acidic and on the warm side out in nature is because it's not a very good competitor. So this is its niche. It's figured out how to live there very nicely, thank you. And other organisms basically are saying, fine, that's yours, take it. Okay? Because it would have been perfect, it would be perfectly happy out in a lake here. But it doesn't. It's not found here. Here's that iron spring I showed you a moment ago, but a close-up, and you can see the Cyanidium condensing on the stone there. They're living off the steam and the few drops of water there. So as soon as it's cool enough for their chlorophyll to stay intact, there they are. So let's look at the high pH for a moment. Let me take you to the Rift Valleys in Kenya. What's happened is you've got the high salinity and the high pH because what's happened is that there were inland lakes there and inland oceans that are dried up. And as they dry, the pH is going sky high. And you also have a certain amount of salt. So here you see some of these lakes, some of the hot springs. That area there, well, I don't think I should have the other picture, about 60 degrees or so. And yet, well, actually some of it's 60 with the hot springs, some of it's more like 40. You can see fish really pushing the limits of temperature there. You can see these Maasai women using it to wash their clothes. Multi-purpose field site. Up in the top there are those flamingos in Lake Bogoria, which I showed you a few moments ago when I showed that whole picture of the pH. And I mentioned that there were cyanobacteria in there too, and you can see in the footprints of the flamingos, they're the green cyanobacteria, which they're living off of. So those are clearly in there at pH 11, 1.5, and so on. And then down here you see in Hell's Gate, very high temperature area. Also a little bit higher pH, but not these sorts of 11 range. Lots and lots of places there. Roaring Mountain back in Yellowstone, getting very low pH environment where you've got these organisms living in steaming acid and they're actually eating away at the mountain. New Zealand, here's a hot spring. Whenever I hear people talk about Darwin's warm little pond, I always picture this picture. And it's just surreal. You can see the steam coming up and the really intense blue of the lake being reflected in the steam. Then over on the right, there's a picture of you get hot water going over these rocks and these beautiful algal filaments that can deal with the very high temperature there. So let me take you to Bolivia for a moment. Bolivia is now in the right location. We started going there a few years ago because of the very high UV flux. What you see here is a satellite image that's been processed. So it shows you a snapshot from a particular day, in this case November 8th, 2004, of the UVB flux on the Earth. What these satellites do is look at the ozone. There's one up there right now, in fact, that does total ozone every day. So you get a snapshot and then it's recalculated. And you can see the pink is where should be the highest UVB, and that's right in there in the Altiplano. So it's not only high UV, once you're up at 15,000 feet, it's low oxygen. You notice it, your body notices. Very low precipitation. It's a desert up there, very cold, particularly at night, windy, because there's no moisture in the air to hold on to that temperature. These pictures are just for fun to give you an idea. When you're going up there, it's a totally different operation. That upper left picture just shows the church in San Pedro. It's the last border town in Chile before you go into, really, that area of Bolivia is almost like no man's land. Right below that picture of the church is the picture of the border crossing into Bolivia. So, I mean, we're talking the middle of nowhere, literally. And as I mentioned, I went up there twice last summer, which was the middle of their winter with 2 different film crews. And the first one was National Geographic. They filmed me driving across that border in the snow 8 times. So now I sort of hope that that picture will be in there as soon as that special's out. If it's before the end of the semester, we'll talk about that. We'll show that. That one's on the Origin of Life. So once you get up there, you get all these wonderful mushy lagoons, different water chemistries, different temperatures, all sorts of odd things. Here's Laguna Blanca, where we've done a lot of work. And what you see on the right, it's just kind of for fun, there were some UV boxes a colleague of ours put up, and the organisms that were under UV opaque are now paler than the ones that were under the UV transparent plexiglass for a year. Again, it's probably because they're producing pigments that protect them from the UV, and you don't need to do that. I threw those in for another talk. That's me filming last summer. I mean, it gets cold and miserable, but you know, it looks so great when you see it on television. You feel like it's real, you know, Indiana Jones type. But anyway, that was all that. And we went out to, we've done some work at Sol de la Mañana. This is also up in the Altiplano, but you can see it looks totally different. This is boiling, steaming mud. It's actually not dissimilar to areas of Yellowstone, but the difference is you don't see any boardwalks, you don't see any rangers. It's a little bit easier, we found, dealing with the permitting and so on, and there are, of course, other aspects to it that are very unusual, and so we can go up there. Laguna Colorada, red, everyone knows here why it's red, high salt, no great secret. And so we've done a little work up in that region too. These harsh environments, you tend to see flamencos. So they're the flamencos, again, this intense red. Turns out high salt, high borate as well. Sorry, I went backwards. Australia, why do we go there? Well, one good reason would be to go to Shark Bay. As I mentioned the other day, that's a great place for these Precambrian type stromatolites. But the other reason we've been there before is because there's this wonderful little place in the Flinders Ranges. And I put this series together a couple of years ago because I went there during the middle of this course. And as usual, I felt bad that I missed a week. So I put this together. What you do is you drive. Has anyone here been to Australia? Okay, got 1 or 2. We started off from Adelaide and drove north, and it doesn't look like much when you look at this whole map of Australia to get to Argarula, but it's a hike. It's a couple of days of driving. You start off and you're on a highway, and la-di-da-da-da, it looks like any other place, except you get emus running around and dead kangaroos on the side of the road. And this was absolutely my favorite. This was a restroom, and I'll show you what that sign says in a moment because I think it's so hysterical. And then you're up around the eucalyptus that actually belong there instead of the transplants that we see here. And eventually you get to this nice little resort, and from there it's only another few hours on what they call dirt roads. I would call dried stream beds. There's a linguistic difficulty there, and you end up at this little pond. I'll show you in a second. But here, that's what that sign says in the restroom. Snakes watch your kids. That's a perfect example why punctuation is important. You can just imagine the snakes watching their children out there. Okay, so that's why we're there, is for Paralana. The reason Paralana is interesting is not because there are people there in orange shirts, but the reason is tied in with the orange shirt people, as we call them. It's because it's near the Beverly uranium mine. There's actually uranium in this area too, and so this Paralana Springs has little bubbles coming up from the bottom that are full of uranium and radon and radium. So here these organisms are getting hard radiation, not to mention the harsh Australian sun beating down from the top. So they're getting a double whammy, and yet there's plenty of goop in there, there are plenty of microbes, all sorts of wonderful things, and we got a proposal in to go back and spend some more time there. Wonderful stuff our colleague Roberto Anatori in Australia has been doing, and we've been fortunate to join in on. Aerobiology. This is a picture I took over Niagara Falls, kind of fun. Aerobiology, I think, is one of the great frontiers that we've been ignoring, and we now at Stanford are starting to really tackle. And when I say we at Stanford, I mean between the people from our class and the wonderful people over in Aero Astro, some of whom we have here, are starting to look at these questions of the bio-launch program. But if you think about it, going up in the air is a real problem. You've got the desiccation, and we've talked about that as being a problem. Temperature, ultraviolet radiation goes up, gases start to change, and therefore you tend to get sterilization. As in a lot of microbiology, Pasteur did the early work, a lot of military environment, much of it's under lock and key in Fort Detrick, but you can imagine the real problems if anyone starts to get involved in germ warfare, which is aerobiology. Today we're concerned very much with disease spread in hospitals through the air, preservation of monuments like the Lascaux Caves, and so on. So what I did was I put together this little 2 or 3 slides trying to show a little bit about what we've done in bio-launch so far. This will just show you one of our launches. I don't have the sound on, but it's mostly people cheering. But what we have is a whole payload at the bottom there of experiments, organisms and DNA and so on that we've sent up with a spectroradiometer to look at what the solar radiation's like and what happens to these organisms as they start to go up. We go up to, the record so far is 95,000 feet, which is about 3 times as high as an airplane. Hopefully there we go. There's a picture from the balloon. Isn't that amazing? You can say ooh and ah now for the tape, the iTunes. Isn't that amazing? I mean, you see the curvature of the Earth. It is just, it psychs me out every time I see it. Unfortunately, there was a water bubble on the camera lens. So anyway, hopefully we'll talk a little bit more about that in a couple of weeks, but it's just an amazing program. So basically, this class now has access to space. I mean, doesn't that just make your spine tingle? We can do stuff in this class. We don't have to just sit here and talk about it. And so I will do a quick commercial announcement and say that the last couple of years we've had a couple of students from the class who've wanted a second semester. And so what we've done is independent reading. So 2 years ago we did one with evolution, and last year we did an aerobiology one where we did some sort of literature survey and tried to learn something, but we all flew on one of the bio launches, so we all worked on experiments most of the semester. So it was just an absolute blast. And if enough of you want to do something like that again, either with bio launch or with something else, I'm certainly open to ideas. So no tests, no anything, just it's just something that you really are fascinated with, and we have fun. So that's the profile of the first launch. We have a lot of these others. You can see we're way up there, we're local, we're a lot of fun. But we're talking about these extreme environments in the Earth now. The early Earth was even worse, and this should look absolutely 100% familiar if you were awake in class on Tuesday. Early Earth, wham, the sun. We've got the radiation, the UV coming out. We've got the atmosphere, which was grossly different when life arose, particularly not having an ozone layer, so we've got very high levels of UV. We've got all those nasty impactors, rotation of the Earth issues. Remember that late heavy bombardment period? We had water chemistry issues. We had all sorts of things that were different. So there were many more extreme environments, and certainly ones that were very different in the early Earth. This is the same picture I showed the other day, except I threw in a picture of the Earth and the Moon from Galileo behind it. But this is to remind us that we've been blocked from radiation below 200 nanometers all of our existence because of the CO2, but it's that UVB and a little bit of UVA, certainly UVC and the UVB primarily, that have been blocked by the ozone layer. And they're not completely blocked, otherwise we wouldn't be getting skin cancer today and so on. But a lot of it has been blocked. And so that really ends the main portion. And if you want to stay, I've got about 5 more minutes worth just sort of to tell you how you can get some money and so on. So I'll just blast through. If you really have to dash, just do it quietly. So we'll show you what's next. Well, we can take some of what we know about extremophiles and apply those to going into space. There's some astronauts having a good time in the space station. We can use them possibly to terraform Mars. We can certainly use them to look for evolution elsewhere. We can use them in biotechnology. There are several companies that go bioprospecting. What they're doing is looking for organisms with enzymes that will operate under extreme conditions. This is just a figure that I put together looking at patent pages a few years ago. And what this shows, there's a couple of these categories of organisms like thermophiles and halophiles, and some of the enzymes that have been patented out of them and used in commercial processes. The favorite one that I love to talk about is one that's used to break down lignin. Now, we know that probably no one, well, a lot of people have computers, but people who are using paper and taking notes, I bet everyone has bleached paper in this room, am I right? Is there anyone who's writing notes on unbleached paper? No, we like using bleached paper. We even use bleached toilet paper. I mean, that's ridiculous. It's totally overkill. But the fact is that we use large quantities of bleach, which isn't great for our environment, just bleaching paper. And it's also expensive if you're worried about the bottom line. And so anything that would cut down the amount of bleach you need to use for paper production is going to be helpful. Well, it turns out there are some enzymes that have been isolated from organisms that live at high temperature and high pH that can deal with a little bit higher pH and temperature than others. And so they're added to the paper pulp to loosen the fibers so you don't need to use as much bleach. And that alone is a several billion-dollar industry. So the mind boggles on what can be done with extremozymes. Wonderful field to get into. And of course, the most important thing for this class is getting back to the idea that this informs our search for life elsewhere. I don't know, is anyone from Iowa here? Okay. Then we can all laugh at this cartoon. Anyway, space is obviously an extreme environment that we haven't dealt with yet. Differences in atmospheric composition, gravity, vacuum, temperature. We're going to talk a lot about that and how that relates to humans in space in the second part of the course. But just to give you a preview, that radiation is going to be one of the big ones because we'll now be outside of the Earth's magnetosphere. And so we're going to get not only much more UV radiation, but we're going to start have to worry much more about solar particle events, solar wind, solar flares, all sorts of issues. But yet we want to follow the water. How much water do we need? Looking at desiccation-resistant organisms gives us some clue. Here's some of the places we're going to visit in the next couple of weeks. Enceladus, Titan, Venus, Europa, and so on and so forth. All sorts of possibilities. I'm going to finish with this, and it's sort of a take-home message. Actually, the last slide will be the real take-home message. But we've talked a lot about individual extreme environments, high temperature, low pH, and so on. But when I started to show you examples from real life, we started to talk about multiple ones, like poor little Sulfolobus that likes to live in boiling battery acid. These organisms that were high temperature and high pH. Well, we don't really know what this three-dimensional or multi-dimensional niche space really is for life on Earth. I've just been giving you the champs for temperature or pH or radiation or whatever. But maybe you can't live at very cold temperature and very high pH. I don't know, I'm just throwing that out. Or, very high salt and very low pH or something like that. And the reason I particularly didn't want to miss this is I'm convinced this is the next step. And this is, the first thing needs to be done is a really good literature search. And so if any of you in the room are interested in doing an honors thesis or a co-term along these lines, I would be absolutely delighted to supervise it. Because I think this is the next thing. I know there was a colleague in England who was starting to put together tables of this, and then he retired because they have to retire much earlier there. And so I now have this bug that we need to start looking at multivariables to really get an idea what the multi-dimensional niche space is for life on Earth. So this is really more the concluding slide. And this is also up at around 15,000 or so feet. This was an Inca ruin. I thought, this isn't so bad. Look at it. It looks sort of like a nice Palo Alto home, nice big footprint. And then I found out that was the llama pen. The little tiny things were the human habitations. Anyway, that's neither here nor there. Anyway, the point is that extremophile, the fact that you're an extremophile is tied in with the fact that we're based on organic carbon, and therefore, to some extent, it's going to be location independent. What I've told you today should apply to life anywhere in the universe should we find it elsewhere. So it actually was worth coming today, because this is something you can apply when you go visit, CLIES 581c or whatever. You'll know something about what the limits will be there, or at least what the difficulties of life will have to overcome. This history of extremophile research, many threads that are coming together, from biotechnology to food preservation, all sorts of things that have come together, and now we'll be able to exploit in the field of astrobiology, and in fact have helped the field of astrobiology no end. This envelope for life is far broader than we could have imagined. I'm going to flatter myself to think that life, what we know about life in its extremes now, is far more than you could have imagined on Tuesday when you left class, or even when you came in here 2 hours ago. It's far broader than anyone could have imagined 30 years ago. And I keep updating this lecture because I'm constantly giving things like this, sometimes once a week. The last 3 weeks has been once a week. And things change. We get someone else coming out with a new record, or a new organism that's a record holder. And so it's just been an unbelievable field to be aware of. Some of these adaptations are biochemically simple and convergent. Some of them are probably very difficult. And so, again, that helps to inform us whether it's likely to happen again and again and elsewhere. And I'll just leave you with this kind of fun little quote from George Bernard Shaw: "The reasonable man adapts himself to the world. The unreasonable one persists in trying to adapt the world to himself. Therefore, all progress depends on the unreasonable man." And you can just put the word extremophile in there instead of unreasonable, I guess.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.